Hello, my name is Anthony De Maria, and I am a biochemistry student here at the University of North Texas. Today, I'll be going over my research as a McNair scholar, which is over coagulation factor V deficiency in developing zebrafish. So, some background. What is coagulation? Well, coagulation is the activation of platelets and fibrin to form a blood clot. And a blood clot consists of platelets that are stuck together by this protein called fibrin. And this protein forms a net across the platelets to keep them stuck to where the site of injury is. Without fibrin, the clot would dissolve or float away from the site of injury. So how is fibrin activated? Well, fibrin is a protein that is activated by the coagulation cascade, which is a system of interacting enzymes that flow in your blood that activate each other, eventually leading to the activation of fibrinogen, which makes fibrin. And you can see it here depicted on the right. And it has two pathways, the intrinsic pathway and the extrinsic pathway, which meet up into the common pathway. So how does factor V fit into this? Factor V is part of the common pathway, sitting there in the middle, and is an important cofactor for activating thrombin, which thrombin is extremely necessary for activating fibrin. So why am I investigating factor V and why in the zebrafish? Well, the zebrafish is an ideal model for hematology research. One, because it has a lot of orthologs for human genes. 71% of human genes have orthologs in the zebrafish. And the zebrafish also has transparent tissues and develops very quickly and forms and gives a lot of eggs as well. So it allows for very quick generation times and screenings with this model. And for the fact as to why I'm investigating factor V, well, factor V has been previously shown in research in literature to be a very important developmental gene along with thrombin. An observation that was made in factor V deficient mice was that they would experience lethality in the embryonic stage, which is very weird, very rare for a coagulant factor to have such a big role in the embryonic stage of an organism, especially if it hasn't exhibited any trauma, experienced any trauma. So what are the objectives of my research? One, it's to propagate an F2 generation of factor V knockout mutant and confirm the genotypes of that knockout. Two, it's to characterize that mutant, and that can be via time to occlusion assay, identifying the lethality time frame, and also observing any morphological abnormalities that are present in the model. And third is to identify possible developmental genes that are associated with factor V via RNA-seq. So the methods. First, what we did was purchase a knockout factor V mutant from ZIRC, which is the Zebrafish International Research Center. And we then crossed that with a wild type zebrafish, which is just a zebrafish with no mutations, forming the F1 generation. And then we did another series of in-crosses of heterozygous mutants with each other to form the F2 progeny. Now, after the F2 progeny is produced, we genotyped that progeny to ensure that the mutation was transmitted to that generation. The whole point of making two generations was to ensure that first, the mutation is stable and is second, that it's transmissible. Third, after doing the sequencing, we'll be using the subsequent generations of in-crosses and growing them into adulthood and observing from the third day that they're fertilized or third days post-fertilization, observing them to see when they experience lethality, if they have any abnormalities, and using them for assays. In the fifth day, after they're fertilized, the heterozygous in-crosses are subjected to laser-induced injury. And there we record how long it takes for them to clot based on injury with a laser. And once we've developed enough of the progeny and measured when they experience lethality, we'll be sacrificing some of the larvae prior to when they experience the most lethality to see how their RNA expression is changing to result in that lethality. And we'll be sending those samples to UT Southwestern for RNA-seq. So my results. First step of the project was to first confirm that the mutation was transmitted to the F2 generation. And here on the right, you have chromatograms of the wild-type heterozygous and homozygous progeny of the F2, factor V deficient mutant. And here, as I've underlined, is the codon of interest. So on the top chromatogram, you'll see that the letter CAG, which is a codon which codes for a normal amino acid. But on the bottom chromatogram, for the homozygous, you'll see that it says TAG. So it changed the base, there was a point mutation that changed the C to a T, making a tag codon. And tag, if you don't know, is a stop codon. So this mutant will have a premature stop in the translation of this protein, resulting in deficiency. And in the middle is a heterozygous chromatogram. And just by looking at the letters, it seems like there's nothing changed. But if you look at the graph, you'll see that there's two overlapping peaks of different colors. One is a blue peak, which is for the C, and one is a red peak, which is for the T. So that shows us that it's a heterozygous, meaning that one chromosome is a healthy copy and the other chromosome is a mutated copy. So after confirming the genotypes, we did a laser-induced injury to the larvae at 5 days post-fertilization. And here on the right is the results of that induced injury. And you'll see that we have 3 distinct distributions of bleeding times for the wild types, for the heterozygous, and for the homozygous. For the homozygous, it took more than 60 seconds for them to clot, if they clotted at all. And the heterozygous, they usually clot around 25 to 50 seconds. And the wild types clot pretty quick from 18 to 22. So what are some morphological abnormalities that we observed? So here we have images of the factor V deficient progeny at 5 days post-fertilization. On the left, you have a wild type, which is just a normal larvae, and figures B and C are homozygous deficient larvae. And you'll see that they have some internal hemorrhaging, especially going on inside the pericardium of the zebrafish, which is some very interesting results. Then we were also measuring the lethality that the mutation would cause in the progeny. So over a series of 24 days, I measured the survival of the heterozygous crossed progeny compared to the wild type progeny. And here is a Kaplan-Meier survival curve, which shows that there's a significant survival deficit of the heterozygous crossed progeny with a 95% confidence. And you'll see that the blue line, which is the wild type, they stay at a relatively stable survival rate of 80%, while meanwhile the heterozygous crossed go down to around 35% survival at day 24. So what are the results? What is the takeaway from this study? First, the Sanger sequencing and time to occlusion data confirm that the presence of a stop codon in exon 2, a factor V, so that results in the knockout of the gene in a bleeding phenotype. So we confirmed that with the laser injury. Later, we saw that the homozygous larvae frequently displayed hemorrhaging in the pericardium as early as 5 days post-fertilization, which is a new phenotype that hasn't been observed in literature before. And also, we saw that the mutation is recorded to be considerably lethal in the heterozygous cross progeny, especially between days 12 to 17. And we're in the process of replicating this data to confirm and even pinpoint exactly what day is the most lethal for this mutation. So what's the significance of this project, and what's the future direction? The significance is bringing greater understanding to factor V deficiency in developing zebrafish. There are very little studies which use the zebrafish model for factor V studies. There's only one other a paper. So this will be definitely bring more understanding to the early effects of factor V deficiency in zebrafish. And it also displays agreement in the early role that factor V plays in both the zebrafish and mammalian model, because as I mentioned before, the mouse also was identified to have a very early lethality in the embryonic stage when it was deficient from this factor. And this data also supports that the zebrafish model as an ideal model for developing and testing treatments for factor V deficiency in humans, such as factor V Leiden and deficiency, which are two different blood disorders that affect humans. Around like 1 in a million humans have that disorder. And the future direction of this project, well, the project is still not wrapped up. The third aim of the project, which is to conduct the RNA expression analysis, is still underway. So the next step of this project is to sacrifice larvae at 10 days post-fertilization and analyze their RNA expression and identify any differential genes, especially in development, that may be occurring. And lastly, I'd like to give some acknowledgements to the co-authors, my co-authors in this study, Sanchit Dinoja and Dr. Jagadish Warren. They've been amazing mentors for me and have allowed me to conduct this research. And also a special thanks to the McNair staff for their endless support. And thank you. Thank you for watching.